It is my great pleasure to welcome Dr. Magdalena Walkiewicz to speak to us today. She's coming to us today from the National Institute of Allergy and Infectious Disease in Bethesda, Maryland. Dr. Walkiewicz is no stranger to the Upper Midwest since she did her undergraduate training just up the road at the University of North Dakota. And while there, she often visited Minneapolis. Going on, she completed her PhD at Rice University, where she was the 2012 winner of the George J. Schrupfer Award, recognizing her outstanding PhD thesis in biochemistry and cell biology. Following this, she did additional postdoctoral training at Rice University and then subsequently did a fellowship in clinical molecular genetics at Baylor College of Medicine. Since 2017, she has been the NIAID, since 2017, she has been at the NIAID, where she leads their centralized sequencing initiative. This program provides an example of the transformative power of broadly applied genomic sequencing as a tool for clinical and translational discovery. Please join me in giving Dr. Walkiewicz a warm Minnesota welcome as she tells us about her work and that of her team.
Well, thank you for this wonderful introduction, and it's just such a great pleasure to be here this morning with you. And I am really, really thrilled to tell you about our experience over the last 4 years of genomics in patients with immune system dysfunction, and then just what we have learned from these patients and what kind of exciting findings we detect in them and how they impact their management and clinical care. I have nothing to disclose today. So what I have thought about doing today is give you a brief introduction to the centralized sequencing program. We have graduated from initiative to program now. It's been 4 years and we thought that it's time to make that name change. I'm gonna spend a couple of slides on program metrics. I've prepared 4 different case reports for you today, and I also have a few slides on new disease gene association. I know that this is pretty ambitious. I am aware of the time constraints, so if I don't get to discuss the new gene disease association, I'll just skip to the closing remarks so we can discuss some of the future directions. And the objectives for today are to identify challenges that underlie setting up clinical genomics research programs, to discuss some of the approaches that we have taken to address these challenges. And then I really want to spend the majority of my time to think about discussing the benefits of unbiased genomic approaches to molecular diagnosis. I think I really like to start my talk by introducing you to my core team. And it is just such a pleasure to have these people out there and discuss them with you, as I am representing their work in this talk as well. When we started 4 years ago, it was just the 4 of us, Morgan Similak and 2 other individuals who are not on this slide anymore because they moved on to other positions at the NIAID. And we have grown substantially. This is the core team that we interact with every single day, and that drives the work forward. We have 3 molecular clinical board certified or about to be board certified clinical molecular geneticists. In addition to myself, Dr. Raj Ghosh and then Dr. Bryce Seifert. Bryce just joined us less than 2 months ago. We have 5 certified genetic counselors. So Morgan Similak and Jai Yan have been with us longest, and then Mike Setzer, Mike Kamen, and Colleen Jadorsky. And then we also heavily rely on the support of ERDAs, and those are individuals that recently graduated from their undergraduate institutions or studies and are in transition to graduate education. Katie has been with us for a couple of years and it's actually moving on to medical school this summer. Mary has been with us for a year and it's also moving on to medical school. And then Riley just joined our team and it's taking over the responsibilities of our ERDAs. We also have 2 technicians in the Department of Laboratory Medicine that work with us very closely, and they are also instrumental to our program. So just to put our program in a little bit of a perspective, I want to mention that clinical genomics have been really fragmented at NIAID. And when Dr. Steve Holland became the scientific director about 5 years ago, one of the initiatives or one of the programs that he was really interested in moving forward was a centralized sequencing program, where with the idea that every patient that is evaluated at NIAID has the ability or has the choice to undergo molecular workup. And prior to setting up this initiative, there were really some challenges both in the clinical arena as well as the research arena that NIAID faced. And through this initiative or through this centralized program, we were really hoping to address most of those challenges. So there was a real need for both molecular evaluation and consultation with genetic counselors. We really didn't have a good way of timely CLIA diagnostics. And then we really were not very good at documenting the molecular findings that were identified in patients based on research sequencing or research genomics. In the research setting, we suffered what a lot of different research institutions suffer, which is siloed data sets. The data usually lives on someone's hard drive, or and it's not really easily searchable, so that one cannot really look through it and look for new interesting variants or genes or so forth. We also had a lot of genome and genomic data challenges. And what I mean by that is that the quality of data that we were receiving from these various genomic studies was very variable, and one could never really truly rely on the quality of the data in making a decision whether something isn't really found because there isn't a molecular defect, or are we missing something because the quality of the data that we are looking at is really poor. And last but not least, we didn't really have a good way of standardized phenotyping of our patients. And we felt very strongly about that last point because we were hoping that in the future, we could just do a phenotype-driven analysis of our data in addition to the genotype-driven analysis of our data. So to address these challenges, this is when NIAID came up with the program to centralize all the sequencing and introduce that to all our patients that are evaluated in the clinical center. So what that entails is a genomic workup for every patient. And historically, we have done exome sequencing coupled with chromosomal microarray if needed. And the idea was that we would look for primary diagnoses. We would also report secondary findings. And for patients that do not currently have a molecular diagnosis, or we didn't find anything that could explain their phenotype, we would actually issue a negative or inconclusive negative report. The main goal was that these results are then actually entered into our medical record or electronic medical record, or called CRIS at the NIH. And then last but not least, we then discuss all these results with our patients so that they can understand what we found in them, as well as communicate this to their team that they are being cared for at home. And, you know, all of that was with the hope that our findings would lead to earlier diagnosis and then subsequently, of course, to treatment and prevention. And I really like to just bring your attention to this little logo that we have underneath here. And I think there were 3 qualities that we were really, really interested in. And that was that we are systematic, that we provide high quality data and analysis, and that we are very transparent in how we do this. And last but not least, we were hoping that the clinical work that we perform at NIAID will actually feed the research work as well. And then the research will then move us forward to more clinical role. So the circle that, you know, clinical work feeds research, and then research feeds back to clinical work. So what I wanted to show you here is just how our program works. And I don't want to spend too much time discussing all the details. What I really wanted to do here is just tell you that when we were designing this program, we were very, very interested in automating and systematizing as much as possible. And then we were also very sensitive to the idea that we will have to provide both tailored clinical and research support to our participants. And what we mean by that, the clinical teams that we work with, as well as our patients. So as I've mentioned, some of the challenges that we faced at NIAID included the quality of the data, as well as the standardized analysis of the data and so forth. And to address that, we came up with this really with this process that I'm actually showing you here in this swim line diagram, where we systematize the entire process so that every patient that comes to NIAID is consented. We place an order in the electronic medical in CRIS. We have a blood draw. The DNA is extracted in a CLIA setting, and this was really important because when we initially started, we quickly uncovered sample integrity issues that we were dealing with. We actually don't perform the sequencing or array analysis at home. When we initially started this program, sequencing was done at CIDR, at Johns Hopkins. We now moved to Baylor, and both sequencing and the chromosomal microarray analysis is done at Baylor. The processing of the data is also standardized. All the data is processed in the same way. Variant interpretation is provided by the board certified clinical molecular geneticist. All the results are CLIA SANGARD and then returned back to patients. By providing this systematized process, we were really hoping that we are addressing some of the challenges that I was mentioning at the beginning of my talk. I also wanted to just spend 1 minute on discussing another aspect of this particular approach. We have grown since the beginning of our program, and now are actually working with another institute, the Mental Health Institute, as well as with NHLBI or some PIs at NHLBI, and we are providing these services to those two institutes as well. So it was really critically important that we can scale up if necessary. And in green, I am highlighting the processes that are easily scalable. We can easily accept more patients to our process and just move that forward. In yellow are the processes that are much harder to scale. So consent is one-on-one. We actually were able to now, in the time of COVID, move from consent by phone to actually electronic consent. We think that variant interpretation, as well as the CRIS report, is also, we have the possibility of scaling it. That just requires a little bit more work. And then the processes that I highlighted in green, in red, is where it's hard to scale. The genetic counseling, of course, is time consuming. Our patients are most of our patients at NIAID are information seeking, so they really like to meet with us and discuss their genetic findings. And our patients are also often suffering through years of chronic disease, so their counseling needs on the social scale are a little bit more intense than maybe what we see in other places or other institutions. And then I just want to point you to this red icon here when we have discussion. I just want to tell you that in the last 4 years, we really have spent a lot of time building relationships with all the clinical teams that we serve. Myself and the other two molecular geneticists have met with various team members on at different times. Some of our PIs like to meet weekly, some of the PIs like to meet monthly, and other groups of people we meet with quarterly, when we sit down with them and we discuss the data for all their patients variant by variant. And this has been really, really critical, because I think it allowed the PIs or our clinical researchers to really understand how we think about genetic information, and then it helped us to understand how the clinical teams think about their disorders. They are the experts, and this discussion part has been really critical in learning about the different diseases, and also just building a really good team and relationship. So last part of the conversation about the program, I really want to introduce you to the interface that we've built to where both the phenotypic data and the standardized phenotypic data and the genomic data live. And I really want to give a shout out to Dr. Sandhya Sarisagar. She has been leading the program. She's been leading the effort on GRIS, and we have had a wonderful collaboration over the years in building this interface that is really allowing us to do a lot of the work that have been not possible if we did not have this interface in place. So GRIS, or Genomic Research Initiative System, holds it's 2 off-shelf products that have been heavily customized towards our needs: Phenotypes and Seeker. Phenotypes, again, is where we store all the phenotypic information. And if we were doing this in real life, this is how this would look like. We enter the phenotype of the patient, again, in HPO standardized terms. We have contact to the person. We have immediate contact to a person, with the person to whom the patient belongs to in GRIS. So if there were any research or clinical questions, we can just go straight from here and email this person about our questions. We include all the protocols that the patients are seen at NIAID for. We really strive to have at least 3 generation pedigree for every single patient that we see at NIAID. And last but not least, a direct link to the genomic data so that we can just access that immediately from the phenotype side. Now, if we were on the Seeker side, which is where the genomic data lives, and we were looking at a certain variant, this is how the data would look like. So we have some identifiers for the family, short description of what is found in the particular patient. So you can see this was analyzed almost 3 years ago, and we identified a de novo pathogenic variant in a gene that explains the phenotype of the patient. In this particular case, we did a trio analysis. So you have the genomic information for the proband, mother, and father. The gene, we have the constraints from broad nomad included in GRIS right here. If this is a known disease gene association, it links us to OMIM. We also have the ability to make gene lists, so we can make our analysis more tiered. The genomic location, if we were doing this in real time, we could take a look at the quality of the data by looking at the reads here. Then the nomenclature for the particular variant, access to all the different population databases, if again we were doing this in real time, I could show you that for every patient that is reviewed in GRIS, and there's a snapshot of how the data looks like, and what was done and what kind of variants were identified, and whether something is reported in GRIS, whether it was Sanger validated, and so forth. Another aspect of this that I'm not showing you here is that if we were doing this again in real life and clicked on this gene symbol, it would link us out to a lot of different websites that can be useful. For example, UniProt, PubMed, Monarch, and other websites that provide a lot of information in accessing variant pathogenicity and so forth. So this GRIS was also, I mean, the reason why we've designed GRIS is to address the challenge of siloed databases and the phenotyping. So we have the ability to search across the entire database at any time for new gene associations. And this can be done by just clicking right here, gene search. So if I came across a gene that I was interested in looking how many times have we seen it in our cohort, I could just click right here, enter the gene name, and it will pull any patient that has had a variant in that gene with frequency below equal to or below 1%. So that really allowed us to make our data sets available to anyone within NIAID or actually NHLBI and NIMH now, that allows us to search for new disease gene associations. I also wanted to mention that as we move on and we classify these variants, this is the classification that our team provided. And if I ever came across that particular variant further somewhere later, that classification would be visible, and I could easily go back to this particular patient and see what were the different classifications and whether we were reporting this back to the patient. So so as we go, the amount of variants that we need to classify decreases, because all of those classifications that we have done so far are captured within the system. Okay, so I've talked about the clinical approaches that we have taken to address the clinical needs at NIAID. I also want to tell you that since we are a research institution, the research that comes out of this initiative is equally important. And we really hope that as we acquire more patients and more data is analyzed, we'll be able to provide new disease genes associations. We are also interested in polygenic risk scores for our patients. And last but not least, contribution from structural variants and mosaic changes. On the clinical, on the return of results, a part of our program, there's we work very closely with NHGRI, which is actually leading a lot of efforts in understanding of genetic testing results. So we have a survey where we survey our patients for their understanding of negative or inconclusive testing reports. We are interested in understanding what do patients actually do with the information, especially in the light of secondary findings. Do they receive that information and actually don't do anything, or do they actually go back to their primary care physician and seek additional help? And then last but not least, we disclose a lot of secondary finding results to minors, and we are interested in understanding how do the parents perceive that information. Often they benefit from it because these variants are often inherited from a parent that either is aware of a family history of certain disease or actually it isn't, and it might be some important medical information for a certain family. So those are kind of, this just kind of gives you a background of why we came up with the program we came up, what are the clinical benefits of this program, and then the underlying framework for research that we hope to continue based on the data that we are collecting over the last 3.5 years. So just to give you an idea of where are we. Since 2017, we've consented over 3,500 participants. We've analyzed data for almost 3,000 of them. Majority of that data were exome because we just moved to genomes in this fiscal year. So that includes the 467 of patients that we have sequenced thus far this fiscal year. Chromosomal microarray was clinical chromosomal microarray was performed for 649 individuals. And then we also do occasionally Sanger-only analysis for family members when we are trying to understand inheritance or segregation. Thus far, we issued over 2,500 reports. 1,500, a little bit over 1,500 were negative or inconclusive. And then 1,000 individuals had a custom report where a molecular finding of some sort was reported back to the patient. So when the molecular diagnostic rate is often what a molecular geneticist or geneticists discuss. And about 32% of our participants have a primary diagnosis. I don't want to deceive you. This is not here, but with this number, a lot of patients that come to NIAID already have a molecular diagnosis, and they are seen by the experts that deal with certain diseases. So those are not all new diagnoses. I think when we looked at this a while back, it looked like we were contributing another 10% of diagnoses. And a very important point here is that we are very cognizant of the fact that this field moves very quickly, and there are new disease gene associations that come monthly. So our program has put forward a systemized approach to reanalysis, so that we look quarterly at new variants that are reported in NIH GMD, and then yearly or probably every 6 months at in more systematic way of new disease gene associations. And then, of course, anytime a new publication comes out and we become aware of that publication, we go back at our data and look if there are any patients that have variants in genes that have been recently published. For secondary findings, we are about 2%, which is pretty typical of what is seen and published. And then about 2% of our patients have dual molecular diagnosis. This is actually a little bit lower than what other institutions have reported. I think part of it is because we actually do deal with much older patients than what you typically that are typically sequenced or where you would typically think a Mendelian disorder may be contributing to the phenotype of a patient. I think time will show whether this number will actually stay the same or whether it will grow, but we are at about 2% right now. Okay, so this is the nutshell of the program. This is why we are doing this, and some of the challenges that we were facing, I think we were able to address, and based on these different methods that we've put in, I hope that we are actually at a place where we can invite other institutes to participate and actually grow as a program. With that, I'm gonna share with you some of the patients that we have seen over the years and some of the interesting cases that we came across and discuss these patients with you. So this is the first patient has been seen here at NIAID by Dr. Leonakis. And this is a patient that actually is very close to my heart because it was one of the patients that I actually went to see and talk to him and his dad when they were evaluated at the NIH 3 years ago. At the time, he was a 9-year-old male patient. He was from Western Ukraine, and he was seen at the NIH for evaluation of Episet. But we quickly realized that he actually did not seem to have air deficiency. However, the reason why the clinical diagnosis of Episet, or maybe even molecular diagnosis of clinical diagnosis of Episet, was entertained is because on prior sequencing in Israel, there was a homozygous missense variant or that was identified in this patient. And based on these findings, the clinical diagnosis of Episet was entertained for him. I just want to mention that by the time he came to the NIH, he has been seen at many different centers in Europe, in addition to Israel, and there was no unifying diagnosis for this patient upon arrival at the NIH. When we took a closer look at the variant that he was coming with here to the clinical center, we quickly realized that it's unlikely that this is contributing to this disease. This was a very common change. And about 14% of the general population. So the family and social history for this particular individual, he was when he came to the NIH, he was still living with both of his parents. Unfortunately, his mother passed away from metastatic pancreatic cancer a year after he was seen. He lives in Ukraine, West Ukraine, with his father and a 7-year-old healthy brother. And he attends school. There are no pets or no unusual habits or activities. He had very many clinical features that were consistent with EPISED, the classical dyad of mucosal candidiasis as well as hypoparathyroidism. He also had hepatitis, pneumonitis, exocrine pancreatic insufficiency, alopecia, vitiligo, and Sjogren's syndrome. All of these features are often seen in patients with EPISED. He also had some features that were not really consistent with that disease, and they included myopathy. He had a hemorrhagic diathesis at 1 month of age, which was not really clear why. He had some very atypical skin features. He was negative for interferon antibodies, and he also had a normal lymphocyte immunophenotype. So when we took a look at him, there were 3 things that we were thinking about. Either he has an expansion of either he has EPISED and an expansion of a phenotype. He either has, or maybe he has two different disorders that are contributing to his unusual presentation, or maybe he has entirely something else. So very common, he underwent chromosomal microarray analysis, which turned out to be normal for him. And then we actually did proband exome sequencing for him. And at the time at CIDR, we actually also had a SNP array that was a quality control SNP array. However, the SNPs were dense enough that we could actually make some interesting calls from that array. So the SNP array turned out to be normal as well. So when we evaluated his data, we came across this novel missense variant in gene called FAM11-1B. Defects in this gene are associated with poicoderma hereditary fibrosing with tendon contractures, myopathy, and pulmonary fibrosis. This variant was really, really interesting, especially after we performed segregation studies, and we've learned that this change occurred de novo in our patients. So we tested this variant on 2 different samples from the proband and then on the parental samples, and we learned that this was de novo. What was even more interesting is that when we did literature search, we realized that a variant at position 430 was reported previously in another patient as a de novo event with features very consistent with the features identified in our patient. So a picture is worth 1,000 words. This is the patient that was seen at NIH, and this is the patient that is reported in this publication with the variant that is adjacent to the variant that we identified in our patient. And I also want to mention that at the time of the diagnosis of our patient, there were only 4 other patients that were reported at that time. This particular patient and then 3 other patients with variants throughout the gene. In the meantime, there is a group in France that actually studies this disorder, and they were able to publish additional patients. I think we are up to about 20 different families, and the phenotype of the patients is pretty consistent. The patients have very similar clinical features, and as you can tell, the skin discoloration, alopecia, these fingers that are short and stubby, are seen with throughout the entire cohort of all reported patients thus far. So this was a situation where we were able to provide a clinical molecular diagnosis. There is really no treatment for this disease, and since there is just so little known about it, we have put him on a pharmacologic regimen, and he seems to be doing very well. Unfortunately, he has not been able to travel back to the NIH because of the events of last year, but we hope to see him again and evaluate him soon, and just kind of take a look at how he's doing. The next case And I'm just going to take a look at the time. Okay, the next case was a patient that was seen by Dr. Alexandra Freeman, and he was a 10-year-old male that was being seen for evaluation for a transplant. He was actually from Lebanon, and he was born to consanguineous parents. He had 2 older healthy siblings. So he suffered from rashes and eczema, cellulitis, and warts since infancy. He had recurrent pulmonary infections. He had chronic abdominal pain. He had chronic liver disease, and he had a prior surgery at age 8 years for interception. When he came for the evaluation to NIAID, he was severely underweight, and he was just not in a very good health at all. So the reason why we were seeing him is because his DNA was actually sent for research earlier to a lab somewhere in the United States, where we knew that he has DAC8 deficiency, but the molecular defects underlying that deficiency were not known. So he was referred to the NIH for presumed DAC8 deficiency as well as transplant. So similarly to our prior patient, a chromosomal microarray and exome sequencing and a SNP array were performed for him. And actually his parents, because we were considering his parents as possible donors for him. The chromosomal microarray is a much faster test. It came back within 3 weeks, and we quickly learned that the reason why the sequencing did not yield a clinical molecular diagnosis is because he actually has a copy number variant that deletes the distal part of the short arm of chromosome 9. Let me just show you. Here is the data from the proband. Right here, there is a deletion consistent with homozygous loss. Mom is actually wild type, and then the dad is heterozygous. When we zoom in on the data, again, deletion of the distal arm of chromosome 9 in the proband, and then that is heterozygous. So as I've mentioned to you before, our sequencing came with a SNP array that actually became very, very useful here. And I'm showing you just the SNP data for chromosome 9, the deletion in the proband, no deletion in the mother. It actually is interesting. She has another small event right here, and then deletion, heterozygous deletion in the father. So what the other part of data that it's very useful that comes from the SNP array is the allele B fraction. And what this allows us to do is look at heterozygosity for each chromosome or each patient. And what you can clearly appreciate by just taking a quick look, here is the data from the proband, here's the data for the mom, and here's the data for the father. In our proband, when we look at chromosome 9, he has absence of heterozygosity for large portion of chromosome 9. Mom is heterozygous throughout chromosome 9, and then in dad, we are just losing a heterozygosity where he has the deletion on chromosome 9. So this was really, really interesting because this made us immediately think about uniparental isodisomy. And this is a phenomena that often occurs for as a consequence of either trisomy or monosomy rescue. However, in case of our patients, we are not dealing with whole chromosome uniparental isodisomy. We were dealing with segmental uniparental isodisomy. And this often arises from mitotic crossover events or break-induced replication. So you might think that in this particular patient, it didn't really matter because we already knew through functional studies that he was DAC8 deficient. But providing the molecular diagnosis and understanding that this actually arose through a segmental paternal uniparental isodisomy of chromosome 9 was really important for accurate counseling, because the recurrence risk of this disease in other children, if the family chose to have them, would be really, really low. And from the perspective of a clinical molecular geneticist, having a finding like that just makes your job so much more interesting. So that was really, really an interesting case that we shared with the family and of course with all our clinical teams. So let me move on to case number 3. This is a patient that has been followed at the NIH by Dr. Zerbi, and I just want to give credit to my this molecular geneticist Raj Ghosh, who actually reviewed this case. So this was a male, 13-year-old at the time of this analysis, and he actually had a known diagnosis of X-linked CGD. This is the situation where we often see patients that that come to us with a certain diagnosis because we have clinicians that are experts in treating certain disorders. And of course, due to his X-linked CGD, he had frequent infections. But he also had failure to thrive and very poor weight gain, as well as some behavioral concerns, including ADHD, issues with executive functioning, and then also poor organization skills. His pedigree is largely normal, with the exception of the maternal grandmother, who had some psychosocial issues, depression, anxiety, as well as eating disorder. And I just want to spend a second discussing the failure to thrive and poor weight gain. Our patients suffer from failure to thrive and poor weight gain because they are so sick a lot of times, and they don't just they either cannot hold their nutrition, or they just have trouble eating, or they are just so sick and in a hospital that their growth is not typical. So it was really hard to understand whether his excessive failure to thrive and poor weight gain were just the consequence of CGD, or whether there was something else underlying that particular phenotype. So again, same molecular studies that most of our patients have. He had a chromosomal microarray that was normal, and then we actually performed proband and mother exome sequencing. So we did a dual for this particular pair, and we identified the CYBB variant that was underlying his X-linked CGD. It turned out that this change was de novo, mom was negative. But what we also found was a missense variant that we classified as likely pathogenic in OTC. And this is a urea cycle disorder gene. And patients that have defects in this gene often they are the classical patients that present very early in life and have a very severe disorder, but there are also these patients that have attenuated phenotype. And this particular variant that we've identified in this patient, we think it's hypomorphic. And some of these phenotypes that he has been exhibiting in regard to his failure to thrive and poor weight gain can be, and most likely will be, attributed to the fact that he actually has an urea cycle disorder. So when we look back at this pedigree, we know that mom carried this variant, so she was a carrier. She didn't seem to have any phenotypes, and when we look at some of the phenotypes that the maternal grandmother was exhibiting, we are wondering whether this cannot be contributed by the possibility that she's also a carrier for the variant in OTC. This was a relatively recent diagnosis. We are waiting for the mom of our patient to tell us whether the grandmother was tested and what the consequence or what the outcome of that testing was. I think what was really, really important is the mom feeling relieved from having this information, because the family has suffered, the family has really been really worried about the growth and nutrition intake for their son. And they were really attributing this to his early clinical course, where he spent a lot of time in a hospital with different tubes, being intubated and so forth. And the family was thinking that his inability to eat was due to those early events in his life, where we were actually maybe telling them that some of this may be attributed to the fact that he has another disorder, and it's a natural way of his body to reject some of the foods that he was being given. And for those of you, I mean, for those of you clinicians, you know that the shakes and the food that we give to patients with failure to thrive are really loaded with protein, and he was not really tolerating that well at all. So a very interesting case where we were detecting a hemizygous pathogenic variant in the proband. That was a de novo change, so we didn't have to worry about the rest of the family. However, the hypomorphic allele in OTC has been maternally inherited. We think the grandma, we think the grandma has the variant, but we are waiting for this for the sequencing. And then, of course, if she's positive, her other daughter and her daughter's children are at risk for OTC deficiency as well. So I'm going to just tell you about case 4, and then I'm going to skip the new disease gene association because we are running low on time and I want to leave a few minutes for discussion. So I'm going to make this very brief. This was a very interesting case that was actually a patient that Dr. Holland has consulted on here at NIAID. This was a 7-year-old female that has come from Mexico and was evaluated for disseminated cocci. She had both brain and pulmonary involvement at the time of her visit. She also had this really interesting weakness throughout her body, which we were attributing to her brain involvement. In addition, I mean, her brain MRI was pretty impressive, and she had pretty severe hydrocephalus, and the infection was just spread throughout. It was controlled, but we were thinking that all of these features that we were seeing in her are because of the brain infection. She also had a CMV viremia at the time of her being at the NIH initially, and then she also had latent TB. So again, same molecular workup that we do for all our patients, chromosomal microarray and exome sequencing. And when we obtained the chromosomal microarray analysis, there was a gain of chromosome 17p12 of approximately 1.3 megabases. And this is actually the gain that encompasses the critical region of Charcot-Marie-Tooth disease type 1, which causes peripheral neuropathy. This was a de novo change. Both parents were negative for this particular finding. We then analyzed the exome and SNP array, and these were non-revealing. So when we look back at this, when we received these results and we were talking about this patient, we initially were thinking, you know, does this fall into the incidental finding category? And, you know, should we report this back to the patient? She's very young, or is this something that, you know, looking back and actually teasing out her different features, we could clearly say that the weakness that we were seeing in her, which we were initially attributing to the brain infection, is actually its own disease. It may be potentiated by the brain infection because she did have features of Charcot-Marie-Tooth very early in her life. But nevertheless, we actually disclosed this result. And this was a situation where every time we've discussed this case with Dr. Holland or other clinical staff at the NIH, it was really interesting how the genetic actually help us look at the patient from a different angle and say, yes, she has this weakness and that's its own disease. She also has this disseminated cocci, which we don't really understand why yet. And, you know, hopefully in the future we will be able to make that connection, but currently we could only really explain the weakness and where that was coming from. And it was really interesting because when she came back after the initial diagnosis for a year later for evaluation, her phenotype was even more pronounced. She had that typical foot drop and some other features that were really consistent with Charcot-Marie-Tooth. And we also did neuro velocity studies and confirmed the neuropathy diagnosis in her. But the other aspect of this finding that was really, really critical was the fact that some of the drugs that we would consider for treating her infection were actually contraindicated for patients with Charcot-Marie tooth. So, and if we were to give her these medications, they could really impact her clinical course and make it even worse. So in this particular case, you know, the line of what's primary diagnosis, what's an incidental finding were kind of blurred, but the consequence of that finding was actually very useful in treating this particular patient. And not to mention that because having a molecular diagnosis now, she's able to receive some other therapy and treatments to support her neuropathy. And I'm going to skip through the new disease gene association, and I'm just going to close with a summary that I hope that I convinced you that unbiased systematic approach to genomic workup in patients with defects in the immune system can lead to some unexpected and interesting findings. When thinking about our patients, more than one molecular diagnosis is possible. This can result in a blended phenotype or in a situation where two or more distinct phenotypes are possible. And I did not get to discuss the new gene disease association, but by building the database that it's easily searchable across at any time, this now is really a main area of research where we are hoping to make more and more of these the association discoveries. And so I want to share with you some of the future directions that we are thinking and taking. So all our patients undergo genome sequencing. We really want to couple it with RNA-seq to help us understand some of the copy number changes as well as deep intronic variants that we are now able to look at. We want to spend more time on looking at the contribution of copy number variants and structure of variants to the disorders of the immune system. For those of you who see patients with defects in genes that cause diseases of the immune system, the penetrance and the penetrance is so variable and incomplete, I'm sorry, and expressivity is so variable that we really think that there must be modifying alleles that dictate how a certain patient, even within the same family, expresses the disease. And we are really interested in uncovering those alleles. And then last but not least, the contribution of common variants to the diseases of the immune system. Those are some of the directions that we are wanting to take. And last but not least, this acknowledges pretty much a lot of the different teams that we either work with or work for, and they all deserve credit in making the work that I was presented possible. And then, of course, last but not least, patients and their families. And at this point, I'll be happy to take your questions or have a discussion about some of the work that I presented.
Wonderful, thank you so much. We do have some questions coming in. We'll get to as many as we can. The first comes from Dr. Sue Berry in our Genetics and Metabolism division. Do you see ways that this approach can be scaled to other institutions that are not as intensely research-based as the NIH?
You know, I think this is an interesting question. You know, NIH is so different because we don't typically send out sequencing to, you know, I mean, I guess there's no insurance, so the patients come in, and the way we approach this is just in a different way. In an institution like your institution, where you send out a lot of testing, I think there is a way of actually requesting the raw data and building a database where you could definitely utilize that data that you've already paid for and build a research program based on that data. I also think that with sequencing getting cheaper and cheaper and the technology improving, setting this up for your own institution is just going to be something that's going to become more feasible moving forward. I mean, I think, you know, if you do have a large number of patients, and I think right now, as far as patients with neurodevelopmental disorders, for example, I think the diagnostic rate is between 40 and 50%. There is a lot of opportunity for research because you have 50% that don't have a molecular diagnosis, and you know, collecting that data and putting it into a database that is accessible by different parties is probably worth thinking about.
Great, thank you. This question comes from Dr. David Brown. What other molecular analyses, such as other omic studies, can enhance the value of genomic sequencing in defining clinical molecular disorders?
Yeah, so, you know, we are definitely thinking about RNA-seq, and I think we are in a privileged situation because, you know, the disorders of the immune system affect blood, and that's tissue that's really easily accessible. I think it's really harder for neurodevelopmental disorders when we are thinking about, you know, brain and other tissues that you wouldn't really necessarily go after doing a biopsy and studying. I also think that, you know, we briefly talked about mosaic variants. I really think that this is something that we need to look at, and this is yet another genomic study, but I think there is some missing heritability that can be attributed to mosaic studies. As far as other omics, I think something that maybe because I came from Baylor and there was a lot of emphasis on that, but something that I like to think about is also epigenetics. And there are some arrays now that we could definitely think about and look at that and the contribution of that to the different disorders of immune system. Other than that, you know, I think I would probably leave other omics to the different research groups, because it's really hard to just incorporate that into this big program. Maybe as technology develops and as things are cheaper and easier to do with better algorithms to analyze, we'll probably consider moving something to the centralized program.
Great, thank you. This one comes from Dr. Brad Miller in our endocrinology division. He asked, can others access the GRIS phenotype interface outside the NIH?
So not right now. Not right now. We are actually setting up some collaborations. We have some pilot projects that we are setting up. So for example, Dr. Rafaela Goldblack-Mansky has this consortium, or leads this consortium of patients that have certain phenotypes, and we are working on a database that can be accessed from collaborators from outside of NIH, where the phenotypic data and the genomic data would be shared. But this is work in progress. We're not there yet.
And there are, the Phenotypes program and the Seeker programs that the GRIS is built on are available externally. But in the case at least of Phenotypes, you have to pay for those as an institution.
Yeah, so Phenotypes actually lasted funding and it's going to go away. So we are now working on a different interface that we will be migrating all our data into. You know, that's a really great point. I mean, Seeker, we made Seeker work. I am aware of a lot of different softwares that are much better. And, you know, it's really just a situation of whether it's worth looking into something new, or whether we just want to customize and work on what we have. And I think right now we are sitting with the latter, but it is not to say that we will move to something different in the future.
Wonderful. I think we are at time. There was a specific question in the chat, and I will connect the question asker with you so you can share some more of your incredible knowledge. Thank you so much for such a wonderful talk, and happy Wednesday, everyone.
Thank you, Dr. Walkiewicz. It was great. Thanks.
Thank you for having me, and thank you for listening.
Hopefully we'll get to meet you in person one of these.
Yeah, exactly. Have a wonderful day, everyone. Bye.
Thank you. So do I just connect on a different, right? I just go to the, okay, perfect. Will do.
Yep.
Okay, great.